This video is about the citric acid cycle. We're going to start out with pyruvate. And so pyruvate comes from glycolysis and diffuses into the mitochondria from the cytoplasm. Pyruvate goes through a series of reactions catalyzed by enzymes in the pyruvate dehydrogenase complex and loses a carbon in the form of carbon dioxide. Coenzyme A joins with the two carbons left from pyruvate to form acetyl-CoA. Acetyl-CoA is what enters a citric acid cycle. It reacts with the 4-carbon compound oxaloacetate through a condensation reaction to create a 6-carbon molecule called citrate. This reaction is catalyzed by citrate synthase. The citrate is then isomerized by a conatase, resulting in isocitrate. Isocitrate is then oxidized by isocitrate dehydrogenase to form the first molecule of NADH and carbon dioxide. Alpha-ketoglutarate is the final product. Alpha-ketoglutarate is then oxidized to create a second molecule of NADH and carbon dioxide. This reaction is catalyzed by alpha-ketoglutarate dehydrogenase complex to make succinyl-CoA. The cleavage of succinyl-CoA to succinate is coupled with a substrate-level phosphorylation. In this reaction, GDP is phosphorylated to GTP, which then donates its phosphate group to ADP to create ATP, or what we know as energy. The 4-carbon molecule succinate is oxidized by succinate dehydrogenase to form fumarate and FADH2. Fumarate is then hydrated into malate, catalyzed by fumarate hydratase. Malate is then oxidized to form oxaloacetate and a third molecule of NADH by malate dehydrogenase. Oxaloacetate is then used to start another turn of the cycle with a new acetyl-CoA. The overall cycle allows one acetyl-CoA to yield 3 NADH molecules, 3 hydrogen ions, 1 FADH2, 2 carbon dioxide molecules, and 1 ATP. Just a reminder, one glucose molecule is cleaved into 2 pyruvate, so if you want to calculate the yield for glucose molecule, you must double all the products made by one acetyl-CoA. I hope this helped you further understand the citric acid cycle.